Good morning, ladies and gentlemen. My name is Ryan Suter. I'm one of the senior investment managers here at Sharewise, and we're here with David Foster, the managing director of Island Pharmaceuticals. So we're just going to start with a bit of David's going to give us a presentation about Island and how it's been tracking recently, and then we're going to run into a Q&A. So I've got a couple of questions here that will start things off, but if any participants would like to submit any questions through the Q&A function on Zoom, I can pass those on to David. So David, it's all yours.
Very good, Ryan. Thank you very much for the chance. So I'm excited about the opportunity to tell you guys about Island Pharmaceuticals and what we're focused on. Island is an ASX-listed company. We are focused on combating viral diseases. We are a small molecule antiviral drug development company targeting infectious diseases. Some recent highlights that I'd be happy to tell you about. We have a lead program called ISLA-101. ISLA-101 is actually a fascinating molecule that I'll tell you about in more detail. But interestingly, it has activity against a variety of mosquito-borne viruses. So think dengue, Zika, West Nile, yellow fever, all of these viruses for which there are really no therapeutic options. Vaccine availability is very limited, but no therapy. So we think we have a really nice opportunity to help out a lot of people. The market opportunity is huge. Dengue infects around 400 million people a year. It's endemic in more than 100 countries. And again, there's nothing you can do about it, really. So we think we have a great opportunity with our molecule, ISLA-101. We've recently concluded a very exciting phase 1 clinical study. We conducted this in Australia. The data were fantastic, demonstrating that ISLA-101 is safe, it's well tolerated, and that the dose we plan to use in our upcoming phase 2 clinical trial is already achieving blood concentrations that have been demonstrated to be effective against the dengue virus, which is going to be our lead indication. So we're very excited about that. There are some very interesting commercial opportunities we'll get into in more detail. One I want you to pay attention to is called the Priority Review Voucher Program. This is a program that is an incentive program in the United States that I'll tell you about. We do like the idea of expanding our pipeline. ISLA-101 is our program right now. We're looking for other opportunities that meet our strategy of identifying small molecules that come to us with clinical history so that we can move into phase 2 clinical trials as quickly as we can. The World Health Organization has done a great job explaining the problem for us. About half the world's population is at risk of a dengue infection, with 100 to 400 million infections occurring every year. It's a huge problem, a major unmet medical need, and it's not just a tropical problem. I think many times people think that these mosquito-borne viruses are based in the tropics, around the equator, and that's really not the case. In fact, just within the last few weeks, there was an outbreak of dengue in Queensland. We see people going to Bali, Indonesia, getting infected and coming back to Australia. Similarly, in the United States, we're finding dengue showing up in the southern part of the United States. This is a slide I like to include because we update this nearly every time we give this presentation. What you're seeing here with these dots on this map are outbreaks and where these outbreaks are occurring that have occurred within just the last 3 months. So clearly you can see there's the Queensland outbreak that I was telling you about. Likewise, if you look in the United States, the southeastern part of the United States, Florida, Texas, Alabama, all experiencing dengue infections this year. And moreover, the places where dengue is typically endemic are having record-setting years, record-setting bad years. Peru, for instance, is having the worst outbreak of dengue in the country's history. So it's a huge problem today, and it's only growing. And in fact, based on some modeling, looking at models of climate change in the upcoming years, what we're seeing is that many regions of the world are expected to get warmer, expected to get wetter. This is ripe ground for mosquito breeding. And in fact, what this map is showing this is that it is expected that dengue is going to be endemic in these areas that are highlighted by the year 2050, which is just around the corner. If you look now at the northern half of Australia or the southeastern third of the United States, you can see dengue is predicted to be endemic there, as well as in the southern part of Europe. So again, just to remind you, with dengue infections, in contrast to, say, COVID, where you breathe and it's a respiratory virus and you worry about passing it that way. Dengue does not pass that way. It passes with mosquitoes. So a mosquito would bite an infected person and then go and bite an uninfected person. And that's how the virus is propagated from person to person. So whenever you see mosquitoes, that can be a real big problem. And if you live in Singapore, for instance, there's a lot of effort to mosquito control because they're trying to curtail the dengue problem. The challenges, the symptoms associated with the dengue infection it's a broad range of symptoms. It can range from, you may not even know you get it, to headache, to mild flu-like symptoms. You may have a rash. Typically, that's what happens with the first time you get it. The problem with dengue is that there's 4 strains. Typically, what you see is the first time you get infected, you can develop immunity against that first strain. However, if you get bitten by a mosquito with one of the other strains, you can have significant problems that can lead to hemorrhagic fever, and ultimately it can be lethal. So it can be a lethal problem. And again, this is one of the challenges with creating vaccines, because vaccines need to develop immune protection against all 4 strains of dengue simultaneously at roughly the same level. And that is a big, big order to try to fill. The U.S. Army has been working on dengue vaccines for probably 50 years unsuccessfully. There are a couple that have been recently approved. Neither of them is perfect. We think that creates a real opportunity for us with a small molecule that you would take that could be a preventative or a potential treatment. The commercial opportunities, we think this can go in many different directions. We believe that this, on the one hand, looks like the traveler's market, like anti-malarials would be a good example of what this might look like. Anti-malarials that you would take before you travel to an endemic region, and then they can also be used as a therapeutic. That's approaching $1 billion market opportunity right now with generic drugs, which ours would not be a generic drug. We also think the U.S. military is going to be interested in what we're doing. And in fact, we know they're aware of what we're doing. We have a cooperative research and development agreement in place with the U.S. Army. The U.S. Army is giving us some reagents and data out of some studies that they've done, which I'll tell you about in more detail, but they are very interested in our study and want to see the results out of it. We also know that places like Latin America or Southeast Asia, where there are these massive outbreaks of dengue, where millions of people are afflicted every year, we think there may be opportunities there where the governments might buy ISLA-101 and stockpile it so they can disseminate the drug whenever they have a regional outbreak. And finally, I want to come back to this priority review voucher, which I told you about at the beginning. This is a really interesting incentive created by the FDA in the United States to encourage companies to develop drugs for underserved diseases. There are many different priority review voucher programs. I'm going to tell you about the tropical disease priority review voucher. The way this works is if you get your drug first approved on a list of diseases that has been established by the FDA and by the government in the United States, then you get this voucher, and the voucher enables you to get priority review of a different program at the FDA. When you submit your new drug application, you get priority review. Typically, review at the FDA takes about a year. If you get priority review, it takes about 6 to 8 months. So there's a huge benefit to getting drugs approved 6 months faster than they might otherwise have been. And you can sell these vouchers. They typically sell in the United States for about $100 to $110 million. So we think that's a really nice opportunity. We're eligible for that today. We don't get the voucher until we get the drug approved. But once we do, then we would anticipate selling it, which would be a nice outcome for all the investors as well as the company. I'll tell you a little more about ISLA-101. Again, fascinating molecule. It was initially developed, believe it or not, about 30 years ago by J&J. J&J developed this as a potential cancer therapy and ultimately abandoned the program and did not pursue it anymore. But nonetheless, ISLA-101 has been in probably 40 plus clinical trials. It's been in literally thousands of people in clinical trials. It's got a well-known safety profile, but it's never been approved for anything, not for safety concerns, but because it hasn't demonstrated efficacy as a cancer therapy. We came to ISLA-101 because we had seen data demonstrating that it had activity against these flaviviruses, against the dengue and Zika viruses, which we were very interested in. And in fact, we licensed this technology from Monash University. Monash had done some very elegant work demonstrating that ISLA-101 had activity against all of these viruses. They also took freshly isolated human cells, cultured them, and then looked to see if ISLA-101 would block the infection of human cells with the dengue virus. And in fact, it does very potently. These labs have gone on to demonstrate in animal models of a Zika infection as well as a dengue infection that ISLA-101 was protective. And finally, sort of the icing on the cake for me, looking at this preclinical package, was Monash had done all this elegant work. Harvard also was doing work with the same molecule, looking at the same viruses and arriving at the same conclusion. So the biochemist in me is very happy to see that this academic work has been reproduced in multiple world-class labs, giving us great confidence that we're seeing a real effect. And in fact, we have sponsored some additional work looking at the ability of ISLA-101 to be protective against these viruses. And in fact, we showed the same thing. One of the things on the last bullet point on this slide, our dose escalation study I mentioned, we've already completed this study, demonstrating that ISLA-101 was safe, it's well tolerated, and it's going to achieve blood concentrations that ought to be effective to curtail the virus in our upcoming phase 2 clinical trial. Briefly, I want to give you a highlight about the mechanism of action. Again, this is some elegant work done at Monash University. On the left-hand side, what you can see, when a virus infects a host or human cell, it really hijacks that cell and turns it into a virus-making machine. In this case, the virus infects the cell and then creates viral proteins. One of the viral proteins is called NS5. And without getting too complex, the function of NS5 is to cycle in and out of the nucleus of the host cell. And ISLA-101 has been shown to prevent NS5 from getting into the nucleus of the cell. So it targets a host viral protein interaction, prevents NS5 from getting into the nucleus, and it's been demonstrated that that's enough to shut down propagation of the virus. And in fact, the graph on the right is showing you the concentration or the ability, the power of ISLA-101 to prevent that infection. And the data here shows that the EC50, or the concentration that is demonstrated to be effective against the virus, was 0.81 micromolar. And it's an important number to remember. I'm going to show you why in a couple of slides. This is one data slide that I think is really important. This is a survival curve. And what we're seeing across the x-axis here is time after infection. So this is survival of mice that are infected with a lethal strain of the dengue virus. So we're looking at time after infection across the x-axis. Across the y-axis, we're looking at percent survival. So at the beginning, 100% survival. If you look, however, 5 days after infection, if you look at the red line, 100% of the mice are dead, 0% survival. However, if you dose with 1 dose a day of ISLA-101, you get to the green line where about 20% of the animals survive. If you dose at 2 doses a day of ISLA-101, about 70% of the animals survive. So in a concentration dose-dependent fashion, ISLA-101 is preventing death in this otherwise lethal mouse model. Again, these are the data that really convinced me that I thought this was something we needed to be developing. Some highlights from our recently completed dose escalation study. Again, this was a study that we conducted in Australia. ISLA-101 is safe and well tolerated. This was as expected in view of the well-documented, well-known history of the molecule. However, it was very rewarding for us to see that we got the same result with drug that we have made. This is drug that we plan to use in our upcoming phase 2 study. Also, after a single dose, ISLA-101 was demonstrated to achieve a blood concentration already demonstrated to be effective against the dengue virus. We did an additional study that we recently announced where we did some in silico modeling of we have data demonstrating what the concentration looks like after a single dose. What does it look like after we dose for multiple days? And this is what you can see on the right. And what you can see is you see the peaks and the troughs, and the trend of this is moving up and to the right. And what that's showing you is that by about day 3, or you can see on the slide, the 7th dose, we've achieved a level at which it sort of balances out. The concentration, the peaks and the valleys, they sort of balance out. They achieve what's called steady state. And importantly, there's a line across just below the bottom trough there. This is the EC50 of 0.81 micromolar. This is the concentration at which ISLA-101 effectively curtails the dengue virus that I showed you from 2 slides ago. And we can predict that at all concentrations, all doses, that we'll be using in our phase 2 study. It's well above the concentration demonstrated to be effective against the virus already, giving us great confidence as we move into this upcoming phase 2 study. The phase 2 study, we are preparing revised protocol right now. We're going to be submitting this to the FDA within a couple of weeks. We're also going to be submitting to the FDA the final documents to close out this phase 1 study. The plan is to submit the documents to the FDA. Potentially by July, we should be screening for enrollment in our phase 2 clinical trial. The phase 2 clinical trial is a fascinating design. It's called a challenge study. And by challenge study, what I mean is we enroll healthy individuals, and then we infect them with an attenuated or a weakened strain of the dengue virus. This was actually a virus that was initially designed to be a vaccine against dengue 1, and it turns out it made people just a little bit sick. Rather than throwing that virus away or the vaccine away, the US Army said, we could study that in humans and just learn more about dengue infections. And that's exactly what they did. So they've run many people through the study where they're just looking at the infection. And what I can tell you is people get a little bit sick. They feel pretty crummy. They get a little bit of a rash, but they don't have to be in the hospital. They don't feel that bad. It's mild flu-like symptoms. So we reached out to the Army and asked if we could use this model for our clinical trial, and they said yes. So what we plan to do is 2 arms of this study. On the one hand, we want to pre-treat these individuals with ISLA-101 and then infect them and see if we can prevent the infection. So it's a preventative or a prophylactic study. We then, in another cohort, want to infect them first, and then we're going to start dosing them a few days after infection and see if we can treat these people, reduce the symptoms associated with the dengue infection, reduce viral load, viral levels in these people who have enrolled in the clinical study. It's going to be a great study. This is going to be conducted at the State University of New York in Syracuse, New York, SUNY Upstate. Again, SUNY's been running these studies before. One of the benefits of the work they've already done is they're giving us all of the data from the control subjects that they've already run. This is the beginning of our control database. So it's a number of individuals that we don't have to enroll. So saving us tremendous amount of money, tremendous amount of time. And then in exchange for giving us the virus and all of this data, the US Army wants to see our data as it comes off the study. So we're excited to be working with the US Army on our phase 2 study. There's a lot of information here. I'm going to walk through it quickly. This is an example of data that we get out of a challenge study. And what we're looking at is a variety of parameters that include viral levels, virus levels, as well as symptoms that the subjects complain about when they come back to the doctor. So if you look at, just want to draw your attention to this blue trace that peaks and then comes down. This is the level of the virus that's measured in these subjects' blood after infection. And you can see that at day 4, the virus levels go up. Then by about day 11, they start to come down. By day 14, they're gone. This is very reproducible. It's very typical of a dengue infection. Also, you can see these boxes that I've highlighted under the graph. These are the days on which people complain about a variety of different symptoms. In this particular case, and this is data from one subject, they complained about abdominal pain, eye pain, fever, headache, nausea and vomiting and rash. So I just want to highlight, we can get so much data out of this. Now, keep in mind, we're going to do a prophylactic study and a treatment study. You can imagine what I would love to see when we pre-treat individuals with ISLA-101 and then infect them. We'd love to see some of these symptoms go away. The subjects don't get sick. They don't have abdominal pain or fever or a rash. We'd love to see the viral levels decrease, the virus levels decrease here. So these are the things we'll be looking at. Why target dengue first? I won't spend too much time on that. I think we've touched on the importance of a dengue therapeutic and a potential dengue prophylactic, which ISLA-101 has the characteristics of both of those. And again, to highlight the problems with all of these viruses, the numbers are staggering. The problem is huge. There's no effective small molecule therapies for any of these viruses: dengue, West Nile, Zika, yellow fever, Japanese encephalitis. There's no therapies available. Vaccine availability is limited. I mentioned for dengue, there have been a couple of recent approvals. And again, neither of those vaccines is optimal. And I think there's still a significant opportunity for a small molecule like ISLA-101. I mentioned we are interested in building our pipeline. We do have a team that is dedicated to looking at additional molecules that would meet our criteria. For instance, that would be a small molecule program, that would be an antiviral, that would be eligible for a priority review voucher. This is the program I told you about that we find very, very interesting. We're really sort of interested in programs that might have interest to the military or the governments as they're preparing for potential weaponized viruses or something like that. We're also looking for opportunities for possible non-dilutive funding support. We think that that's very important for a program that we might bring in. Just to highlight our corporate snapshot, one of the things I'm really proud of is we have a really tight register. So my top 20 investors control about 75% of the stock here. I do want to point out that we recently concluded a rights offering. We raised $1.95 million at the end of March. Associated with that rights offering, people were able to get a share and an option, a one-for-one share and option. We created an incentive with that option that says if you exercise your option within 3 months, you get the opportunity to get an additional option. The 3-month deadline for that is this Friday, June 14th. So if you're an option holder and you have any questions about how to exercise that option so that you get your additional piggyback option, please feel free to email me at info@islandpharmaceuticals.com. I'm happy to walk you through that. This is another reminder, again, just for those of you who are option holders, to the deadline for that to receive that piggyback option is this Friday. We have a fantastic scientific advisory board. All of these scientific advisors, card-carrying virologists, drug approvals under their belt, deeply involved in antiviral drug development. I won't go through all of their details, but they're a fantastic group. We're happy to be working with them. I do want to highlight some upcoming milestones. It's going to be a very exciting 6 to 9 months for us. I've already mentioned we're looking to start enrolling for our phase 2 clinical trial. That should begin screening and enrolling probably in July. We anticipate dosing beginning in August. Dosing should be complete by around October, with data coming in by the end of this year and into Q1 next year. This is a lightning fast phase 2 study. One of the beautiful things about the clinical trial we've designed and looking at infectious diseases, dengue in particular, is we know within a matter of weeks whether or not the drug works. The life cycle of the virus is very short. So we're going to know whether we have an effect on viral load and these symptoms associated with a dengue infection. So we're excited about the next 6 to 9 months. I think it's a great opportunity for people who are maybe looking at Island Pharmaceuticals today. I think we have a very exciting 6 to 9 months where there's going to be a lot of value created. And with that, I'll pause and take any questions that you have, Ryan. Thank you very much for your time.
Thank you very much, David. That was absolutely fascinating. Now, you did mention, so I'll start off with a couple of questions, and then I've got a couple of questions from the audience. So probably the first one is realistically with the phase 2 clinical trial, what's the timeline from like when you reckon that'll start like actually, occurring, and then when you reckon it'll finish and we'll have results?
Yeah, great question. And I touched on it, but we expect to begin enrolling. So we're going to advertise for people to enroll in this study. That's probably going to start in July. So, I mean, we're a matter of weeks away from advertising where we can enrolling, screening and enrolling those patients, or not patients, but healthy individuals. We think that we'll start dosing them. So pre-treating and followed by the infection with the dengue virus, probably in August. We anticipate there being, again, the preventative arm as well as a therapeutic arm, and they will run back to back. So we'll do preventative first. All of the dosing should be done by October. So it's going to be very fast. We'll be through with that. And then it's a matter of collecting the data, looking at the blood levels of the virus. I think data starts coming in by the end of this calendar year, which is literally 6 months away. We're going to wrap up the full data package in Q1. And this is an important piece to highlight. We'll be a company with phase 2 data in Q1. I mentioned the options, right, associated with this last financing. The expiration date on those options is March of 2025. After we have phase 2 data. So I think the timing on that works out pretty well for people.
Okay. All right. Well, thank you very much for that. And obviously, it seems like your contract or dialogue you're having with the US military is very expansive when it comes to pipeline building. Is there anything else, any other potential partners that are noteworthy, whether there are other kind of military organizations or potentially in other countries?
Yeah, so you're right. So we have a great relationship with the U.S. Army and the United States, obviously, and we're doing this phase 2 study in collaboration with them. They're giving us data, they're giving us reagents. They want to see what we're doing. We'd love to work more closely with them. We also spend efforts looking in geographies where dengue is endemic. For instance, I just came back from a partnering conference in Singapore where I was able to meet with some regional pharma companies, frankly, companies people may not have heard of, who are very interested in our program. You might imagine if you're in Singapore, dengue is a big problem for you, and you think about it every single day. Likewise, if you're in Brazil, and I spend a lot of time talking to people in Brazil, a lot of those conversations, Ryan, today are making sure these people know who I am, making sure they want an update from me every time I come back to the conference.
Yep.
And so I stay at the front of their mind. I think as we start getting into phase 2 data, starting to see some of these phase 2 results, I would anticipate partnering conversations start happening in more earnest when people get really excited about that.
Okay. All right. Well, that's really positive news. And then on the basis of the phase 2 trial results performing well or performing as anticipated, what's next for Ireland?
Yeah, great question. So we've sort of set this trial up, this phase 2 trial, to give us a lot of data. Looking at dengue is looking at ISLA-101 as potentially a preventative or prophylactic molecule, or as a therapeutic molecule. The reason that's so important is the trial that we do after this likely is going to be conducted in endemic countries, will likely be in Southeast Asia as well as Latin America, and it's likely going to be a therapeutic study. So that's what I think the future looks like. That's all subject to conversations with the FDA, but the way I see this playing out is following the phase 2 study, we'll talk to the FDA, and probably it's going to be a therapeutic study in endemic countries.
Okay. All right. Thank you for that one. So those are all my questions covered. I've got a couple more from the audience. So first question here is there much non-dilutive funding around the world for programs like this one?
So yes, there's non-dilutive money. I'm primarily familiar with pockets of money in the United States government where there may be interest. And in fact, we have received sort of indirectly already about $625,000 US to pay for this program. This was just about a month ago, we announced that there was some US government money that was allocated to do some sort of bonus experiments with our subject samples out of our clinical trial. We asked the government if they would be willing to reallocate that money to pay directly for our study. And they said yes. So we've received, not only are we partnering with the Army to conduct this study, but we've now gotten 625,000 US to pay for the study. So there is some non-dilutive money there that we've already been the recipient of. There are other agencies within the US government and around the globe that we're investigating to support programs like this.
Okay. All right. And on the back of that, so I've got another question here. If you sell the PRV, does the company lose the intellectual property for the molecule, or would the company, would the company be stripped of the asset molecule for cash in that sense?
Yeah, that's a great question. The priority review voucher is completely, we are the recipient of the PRV is given to that person who gets the drug approved based on approval of that particular drug. But once you sell the PRV, it is entirely separate. So the idea would be we would keep ISLA-101, all of the IP, all the commercial opportunities associated with that, but we would just sell the PRV. The person who buys, or the company who buys that voucher, they can use it for anything. They can use it for their next cancer checkpoint inhibitor or whatever they want. But it doesn't, there's no negative associated with our program when we sell that PRV.
Okay. All right. Well, thank you very much for that. It sounds very promising. Oh, well, look, David, we really appreciate your time. I think that wraps us up on my end for questions. Do you have any kind of remarks or anything you want to leave the audience with?
No, I want to thank everybody for their support in the last capital raise. I would remind everybody, watch what we're doing. The next 6 to 9 months are going to be very, very exciting time for us, and I hope you'll come along for the ride.
All right. Thank you very much, ladies and gentlemen. Thank you very much, David.
Thank you very much. Thanks, Ryan.
Cheers.